Kristin Hegstad. I work at the Norwegian Center for Detection of Antimicrobial Resistance in north of Norway, in Tromsø. And we are also a reference lab for Linezolid-resistant enterococci, or LRE for short. And that is the topic of this poster. So since the first case in 2012, we have had an increase in incidence of Linezolid resistance that we wanted to investigate. So in 2012, the incidence was 0.1, while in 2023, it was 1.2 per 100,000 person years. And so we collected all the Linezolid-resistant isolates in Norway and investigated them by whole genome sequencing. As you can see here in table 1, the Linezolid-resistant enterococci were more or less divided into half and half for Enterococcus faecalis and Faecium, but we had one isolate of Enterococcus gallinarum also. Looking at the Linezolid resistance mechanisms, we see that as many other places in faecalis, OPTRA, the transferable resistance, is dominating, while in Enterococcus faecium, it's the mutational resistance in the ribosome that is dominating. But we also see cases of other type of resistance. When we do phylogenetic analysis and look at this minimum spanning trees here in figure 1, at the upper part here is Enterococcus faecalis, and they are color-coded according to the sequence types that we find. So as you can see on the upper part here for Enterococcus faecalis, there is a lot of diversity in sequence types, and also there are not many of the isolates that are related to each other. It's just a few pairs. However, it's quite different for Enterococcus faecium in the bottom here. There are typically these sequence types that are associated with hospital infections, and there are more clusters also that denotes that they are genetically related. And when we look into the epidemiology of these clusters, we see that we have at least 4 outbreaks during this time period. So then we know that at least some of the rise that we have seen when it comes to Linezolid-resistant enterococci in Norway are due to these outbreaks and mutational-based resistance in Enterococcus faecium. And that is often linked to Linezolid usage. However, we also wanted to have a clearer picture of what's going on when it comes to the transferable resistance. So we did long-read sequencing to look at the genetic context of the transferable resistance genes. And then we see here in table 2 that for the OPTRA genes, a lot of the ones that are in Enterococcus faecalis are chromosomal. And we see that there are specific OPTRA allelic variants that are chromosomally related. However, we also see a good variety of other allelic variants that are plasmid related. And when it comes to faecalis, it's mostly pheromone-responsive plasmids that are known to be quite highly transferable among Enterococcus faecalis. While for Enterococcus faecium, it's a very diverse type of plasmids that we see. We also had a few cases of POXA resistance in Enterococcus faecalis, and 4 of them were actually a new variant that hasn't been described before. So this new type, we denoted POXA-EF, because it's only about 70% identical to the ones that have been found before. And when we looked at the genetic context, you will see here in figure 2, we see that this POXA-EF, 2 of the 4 cases are localized on Rep2 Inc18 plasmids. And they are also together then with resistance gene for high-level gentamicin resistance and a lot of these insertion sequence elements that might also give mobility to the POXA gene. We also had the other 2 cases of POXA-EF, the novel variant, that we think are part of linear plasmids. That's something that we are still investigating. And we also have had other cases like for this Enterococcus gallinarum with OPTRA. We also think this is part of a linear plasmid, but that's something we are investigating still. So we are also working to add metadata to see if we can have more information about where all these strains come from so that we can explain why we have seen this rise. Because the Norwegian surveillance of animal isolates, they don't find any Linezolid-resistant enterococci. So we think that at least some of them are related to import. So thank you very much for your interest in our poster.